which is really important for understanding disease mechanisms. And one of the examples of this is his group's important discoveries on the immune basis of Alzheimer's disease. So Manolis's talk today will be on single-cell dissection of human disease circuitry. Unfortunately, he has a class that he has to teach at 1 o'clock, and in addition to everything else he does, he's also actively teaching. So Manolis has told me he'll try to have enough time for questions at the end. He's also going to monitor the chat and select the questions at the end. So Manolis, thank you very much for taking time out of your busy schedule to speak today.
Thank you so much, Laurie, for the opportunity, and thank you all for being here on these crazy and difficult times. So what I'd like to tell you about today is our work on trying to understand the mechanistic basis of human disease. So how do we translate genomic information into therapeutic insights? And specifically, how do we understand the circuitry of human disease at single cell resolution? And how do we use that to actually enable manipulation? So it all starts with genetics. The promise of genetics is really to understand disease mechanism in a completely new way, to reveal target genes that are sometimes completely unexpected, and reveal new therapeutics through these target genes and enable precision medicine and also personalized medicine. And the story is exemplified beautifully by obesity, where in 2007, the first genome-wide association for body mass index returned a hit in this FTO locus, which was promptly named fat and obesity associated. It used to be named fused TOS O. And this basically gave a molecular basis to obesity. Instead of, oh, it's just simply what you eat and whether you exercise or not, there were genetic variants associated with obesity. The path was therefore open for therapeutics against obesity. Let's fight the obesity epidemic, everybody said. Let's go after these genes. So they looked at the locus and they basically said, oh, wonderful. This is not one of those complicated loci where there are many genes under the lamppost of genetic association. All 89 common variants that are sitting in this linkage disequilibrium block between these two recombination hotspots and that are all co-associated with the disease are sitting smack in the middle of this FTO gene. So this must be the target, everybody argued. And hundreds of millions of dollars were probably spent on understanding the circuitry of this gene, how it acts, what it does, and so on and so forth. And a lot was gained about this gene, but the problem is that this gene, turns out, has nothing to do with obesity, as we showed with Melina Clausenser a few years later. It turns out that the true target genes are actually sitting 1.2 million nucleotides away. How is that possible? What's happening in the FTO locus and what's happening in 93% of genome-wide association study results is that the disease hits do not perturb the protein directly. So none of these 89 common variants is in fact perturbing the FTO protein. They're instead sitting in the first intron and a tiny little bit of the second intron of the FTO gene. And you can see this tiny little exon 2 here and this tiny little exon 3 here do not contain any genetic variants that alter their function. So that basically means that in all of these cases, we actually don't know whether the true target gene is the one under the lamppost. That's simply where the light is, but that's not necessarily where the targets are. So what we need to know systematically in these 93% of GWAS hits is what is the true target gene and where is it sitting? And is it sort of far away? Maybe more than 1 million nucleotides away. What is the causal variant? Because of co-inheritance of these blocks, these genetic variants as blocks of linkage disequilibrium, these variants are all extremely strongly associated with obesity. So that means that we don't really know which of these variants is in fact the causal one. And that also precludes us from understanding what is the cell type of action, because maybe it's this variant and that region is active in, I don't know, the brain, or maybe it's that variant and that region might be active in the intestine or something like that. And therefore, understanding all of these things is intricately intertwined and also is key to understanding what are the relevant pathways and what are the mechanisms underlying these genetic associations. So that's what my team is trying to do, and that's what many in the field are trying to do. How do we systematically understand the circuitry of the genome in order to be able to decode non-coding variants? And non-coding variants are not an exception, they are the rule. This is what 93% of GWAS looks like. So the approach that we take in my group is that we start with human disease genetics across both common and rare variants. So we reveal these regions of association, and then we profile systematically RNA and the epigenome across both healthy and disease samples to understand how are the molecular signatures of disease changing with these phenotypic variation. We then integrate all of the data to predict driver genes, regions, and cell types. And then we validate the predictions in human cells and also in mouse models through multiple collaborations in the space of brain, cardiac disease, metabolic disease, cancer, and immune disorders. So across all of these, we basically then, of course, disseminate the results and start all over again. And the reason why we're working on so many different disorders is because the methods are general, because the same genomic technologies are needed to systematically understand the human epigenome and the circuitry of the genome in hundreds of different human cell types to then be able to infer common methodologies for inferring that circuitry, and then, of course, linking all that with genetics and with validation. So the view that I want you to sort of take out of this talk is that before our methodologies, all we know is that there's a region of association somewhere in the AFF1 gene in this lipid-associated and cholesterol-associated region. But instead, what we have with these methodologies is the circuitry. Namely, we know exactly what are the variants that are associated in that locus. What are the regulatory regions where these variants lie? What are the cell types and tissues where these enhancers, these distal regulatory regions, are acting? Who are the upstream regulators that are responsible for this effect where their binding might be disrupted by these genetic variants? And what are the specific motifs that are getting disrupted in these regions? And of course, what are the target genes that are getting regulated by these regions? This enhancer here targets the AFF1 gene directly, whereas this enhancer here targets another gene, several genes over, and understanding and resolving this can completely change the mechanistic basis of disease. And the work that we've done has basically resulted in the dissection of many loci across the genome, including this work that we published in collaboration with folks at the Joslin and with Melina Klausenzer, who's also a member here. So I'll be talking about this work a little bit in more detail, but the message that I want you to come across is that understanding the circuitry means we can intervene and therefore switch between lean and obese phenotypes. So what I'll be telling you about today is, number one, how do we use reference epigenomes to predict the disease-relevant tissues? How do we combine genetic, epigenetic, and transcriptional variation in the context of disease to understand how these are varying from person to person? How do we do this at the single cell level, combining epigenomic and transcriptional variation at the single cell level? How do we then turn to mechanistically dissect any one of these regions? How do we then understand not just one tissue at a time, but many tissues and the convergence of these functions across obesity, cancer, Alzheimer's, and aging? How do we study these integration across multiple phenotypes, including medical records? And how do we develop methods for high throughput dissection of these gene regulatory circuits in disease? So let's start with the first one. How do we understand the disease relevant tissues and regulators through global enrichments by integrating this epigenomic information? So as part of the ENCODE project and the Epigenomics Roadmap project, we've basically built a reference of the human epigenome. The same way that the human genome needed a reference for research to flourish, we basically looked for a reference human epigenome. But unlike the genome, which is largely shared among nearly all of the cell types in our body, the human epigenome varies dramatically between different tissues. So the Human Epigenome Project basically sought to decipher, number one, what are all of the tissues and both adult and embryonic tissues, and the epigenome, how it changed among them, how it changed between different regions of the brain, between different primary cells, as well as in vitro differentiated cells from embryonic stem cells and iPS cells. And for each of those, we basically mapped multiple histone modification marks that are annotating the function of the genome systematically in order to understand where are the promoters, the enhancers, the transcribed regions, repressed regions, heterochromatic regions, active regions, DNA accessible regions, and repressed regions through whole genome bisulfite. So the human epigenome is extremely complex, and it has many layers of annotation. DNA methylation is acting directly on the DNA. DNA accessibility is basically letting the compact chromatin open in order for transcription factors to bind. And then the histone modifications are by far the most complex, and they are post-translational modifications on the tails of histone proteins that allow you to denote distal control regions like enhancers or proximal control regions like promoters, transcribed regions, and how they are active in each cell type of the body. So we've used that to now start looking across more than 100 different tissues and cell types for how is the epigenome changing in each of those. And what we're finding is quite remarkable. There's an extremely rich dynamic picture of how the genome turns to life across these different tissues and organs. We see that promoters, for example, are extremely stable and they're open for business whether or not the genes are transcribed or not. We see genes turning on and off in a coordinated fashion across specific lineages of differentiation. We see that enhancers are much more dynamic than promoter regions with extremely cell type specific effects, as well as a small subset of enhancers that are broadly active. But this dynamic view also allows us to recognize the targets of control regions. For example, if I have a non-coding region here that is associated with disease, I can say, well, maybe it's one of the genes nearby. Or I could look at where are the cell types and tissues where this region becomes active, and then look for nearby genes that are turning on and off in the same tissues. And what you see here is that the PAX5 gene is sitting 500,000 nucleotides away, but is the only gene in this region whose expression is actually coordinated with this non-coding region, enabling us to link them together. The second thing we can do is systematically ask where are genetic variants of each disorder associated with each disorder, where are they acting? And we can actually use the availability of many different GWAS results to play them off each other and to truly understand where are the loci that are associated with each region, with each trait. So if you look at height, for example, here are all the linkage disequilibrium blocks and the individual SNPs within them. And type 1 diabetes paints very different regions in the genome. Blood pressure and cholesterol, very different regions. And now we can start overlaying the epigenomic annotations of all of those regions in stem cells, in immune cells, heart cells, and liver enhancers. And what we can see is that genetic variants associated with each of these disorders are enriching specifically in tissues that are quite relevant to those disorders. If you look at the larger picture from which this cartoon is drawn, you can see that indeed genetic variants associated with height only enrich in embryonic stem cell enhancers. Genetic variants associated with all kinds of immune traits only enriched in T cells and B cells. If you look at blood pressure, you see only one enrichment across the entire set of tissues, only in enhancers active in the left ventricle of the heart, where the blood pressure gets built up before you pump it to the rest of the body. If you look at cholesterol, no matter how you measure it, HDL, LDL, etc., you basically have enrichment specifically in liver enhancers, suggesting again that non-coding variants acting in liver are playing a big role in the genetic predisposition to high or low cholesterol. If you look at fasting glucose-related traits for anyone who understands the biology of type 2 diabetes, you would expect to see an enrichment specifically where we see it, namely the pancreatic islets, where the beta cells are in fact getting dysregulating, leading to insulin resistance. If you look at inflammatory bowel disease, you basically would expect to see, well, let's put some money on this. Inflammation, I would say immune, sounds great. And then bowel disease, I would expect these digestive tissues. And indeed, that's what we find. So we gained some money. Let's put some money on Alzheimer's right now. So Alzheimer's, I would guess brain. So let's put all our money on brain. And we just lost all our money. And the reason is that brain is composed primarily of neurons, oligodendrocytes, and astrocytes. But the microglia are in fact very depleted in this, they're underrepresented. There's only 5 to 10% of microglia in these brain samples. That's why we don't see any enrichment. By contrast, when we look at CD14 plus monocytes, which are having the same markers as macrophages and resident macrophages in all of the different cell types, as well as, of course, microglia in the brain, what we're seeing is that there's an enormous enrichment for Alzheimer's. That suggests that perhaps genetic variants associated with Alzheimer's don't act in the brain through the neurons and the astrocytes and the oligodendrocytes or the microglia, but they act instead through the microglia. And indeed, this is what we saw when we started sorting different cell types across neurons, microglia, and oligodendrocytes. We basically saw that cell-sorted H3K27 acetylation, again, a mark of distal enhancers, is in fact specifically enriched in the microglia, but not at all in the macroglia or the neurons. And that basically led us to postulate that there's in fact an immune basis of Alzheimer's disease, which we found to be conserved both in human and in mice. And indeed, we found that early changes in Alzheimer's were predominantly affecting immune processes, whereas late changes were predominantly affecting neuronal processes. So we've expanded this work dramatically from 127 epigenomes to 834 epigenomes, from only 54 enriched traits to 534 enriched traits. And we now have a very rich map of where are the different tissues and diseases co-occurring in this complex network. And what you see is that, for example, schizophrenia, neuroticism, and other psychiatric disorders, as well as cognitive disorders of cognitive traits of intelligence and math ability and educational attainment are localizing very much in brain, which is again primarily composed of neurons and other microglia. If you look at Alzheimer's, again, it localizes in these immune cells, along with a lot of other traditional immune traits. If you look at kidney, you see filtration traits that are localizing there. Liver with cholesterol, you see coronary artery disease with heart and muscle and so on and so forth. And you also see these polyfactorial traits that are sitting in the middle, such as obesity and waist-heat ratio, which are actually involving these complex pie charts with multiple different types of tissues being involved. And what's really exciting here is that by being able to dissect the epigenomic landscape of these genetic associations, we can actually start partitioning these very complex traits into simpler parts. So if we take coronary artery disease here, which is actually the most complex and polyfactorial trait, we see that it shows enrichments in many different types of tissues. So liver, coronary artery, thyroid, adipose, and so on and so forth. And what's really exciting here is that if we partition the genetic loci associated with coronary artery disease into the subset that overlap liver enhancers or coronary artery enhancers and so on and so forth, then we see that the corresponding genes that are associated with these loci have enrichments in very different gene ontology categories. That suggests that we can actually take that disease complexity and peel off layers of complexity by focusing in on specific subsets at a time. And when you do that, you also see that the co-associations with additional traits that are comorbid with coronary artery disease are in fact themselves getting partitioned differentially between the subset of enriched tissues. That allows us to now go into individual loci that are associated with coronary artery disease and say, well, hey, this one seems to be enriched with liver and thyroid and so on and so forth, whereas that other one appears to be only enriched in coronary artery. And indeed, when we look at the corresponding loci, we basically are now able to start linking together these non-coding associations to their target genes and then predicting what are the tissues and genes through which they're acting. So we can see here PCSK9 acting in liver in association with coronary artery disease. You can see here eDNRA acting in coronary artery, again associated with CAD. And you can also see this locus here, PLPP3, associated with both liver functions and coronary artery functions through potentially multi-tissue pleiotropy across multiple genes. We've basically built now a website that you can use to explore all of this information. You can look for EpiMap for epigenomic map, and you will be able to find what are the regulators associated with each tissue, what are all of the traits that are enriched in every tissue, and for 30,000 loci that are localizing within enriched annotations, you can basically go and dissect the specific circuitry of these locations. So that's using reference epigenomes that are making the, of course, oversimplification that the epigenome of everybody on the planet is the same, which is, of course, not true. Instead, we can start now asking how is epigenomic information varying across individuals along with transcriptional variation, along with also genetic variation between these individuals, and of course, disease status across those individuals. The goal of this work is to start bridging the gap between genetic variation and disease, which is enormously large, by recognizing the individual tissues and cell types where these variants are acting, the epigenetic regions that are changing in activity in response to these genetic variants, the convergence of these into common gene expression patterns, and ultimately endophenotypes, which are underlying disease. And we can start tracing individual genetic variants and their action through this path. Of course, this also requires being able to resolve how many of these correlations in these intermediate sites are in fact causal. And the reason why this is complicated is that as soon as you have genetics in the picture, the arrows are unidirectional, because a correlation with an A to G common variant that is segregating the population is not going to be due to simply the fact that, oh, I've been smoking a lot and therefore, I have this inherited genetic variant. Yes, of course, for somatic mutations, but not for common genetic variants that are inherited and segregating through the population. Instead, however, when we have enhancer gene correlation, we don't know whether the enhancer drives the gene or the gene expression drives an epigenomic rearrangement of the landscape in that region that also opens up the enhancer. And similarly, if we see a gene going up with amyloid, it could mean that the gene is in fact causing amyloid, or it might mean that the increased amyloid is causing the gene to go up. Or it could be that the gene is in fact combating amyloid. And therefore, therapeutically, instead of repressing the gene, I might actually have to overactivate this gene that is positively correlated with amyloid. And they also might be simply correlated without any causation. They might both be due to an environmental change, which is simply coordinated between the two without any actual relationship of causality. So what we can do now is actually start understanding how to link together these unidirectional arrows from genetics and understand the mediation effect of genetics on these genes and these phenotypes in order to be truly able to infer causality. So as a case study, we basically looked at methylation variation across 700 individuals in the ROSMAP cohort for Alzheimer's that David Bennett put together many years ago. And we've looked at epigenomic variation in concordance with genetic variation and of course phenotypic variation, but also transcriptional variation in those individuals. So the first thing that we can do is ask how much is the genome predicting the epigenome? And can I use that to then start relating to causality and phenotype? It turns out that the genotype is extremely predictive of the phenotype for DNA methylation, that there are 50,000 different genetic loci with these astronomical 10 to the minus 300 p-values, where if you know the genotype of a person at birth, you can actually predict their DNA methylation levels in an inaccessible tissue like the brain at 93 years of age. So that is very exciting because it basically means that I can actually start predicting the methylation landscape of an individual using their genetics. Why is that exciting? Because if I can do this G2M prediction, predict methylation from genetics, I can then start correlating methylation with disease. And if I correlate methylation with disease, this is an MWAS, a methylome-wide association study instead of a GWAS. And that is actually less polygenic because there are only a much smaller number of methylation changes associated with disease, as opposed to individual genetic variants that have a much smaller effect because they're much more polygenic. However, this arrow between M and D is bidirectional if I use observed methylation. If, however, I use this G2M to predict methylation, to only carry out imputed methylation association with disease, the imputed methylation is different from the methylation. It's just a genetic component of methylation. That allows me to now start correlating only the genetic component of disease and therefore get a unidirectional arrow back. This is just a subset of methylation that's driven by genetics. That's very powerful because I can go now back into regions that had no genome-wide significant loci and start predicting new genetic associations through these intermediate phenotypes by combining multiple SNPs to predict methylation, and then seeing if the imputed methylation is in fact correlated with disease, thereby leading to many new additional phenotype associated genes whose expression or whose methylation is in fact driving this significant association. And I can do that through methylation, or I can do that through transcription. I can do that through the combination, and so on and so forth. And looking at transcription, we basically see that across Alzheimer's disease, you see many new loci. You see, of course, many genes in genome-wide significant loci that are shown in purple, but you also see additional loci that are not reaching genome-wide significance without this methodology, but are clearly associated with the disease and capturing a large fraction of the heritability of the disease by combining multiple genetic variants in the locus. You can also find the tissue of action, because we're looking specifically at brain here, and the directionality of effect, whether they're positively or negatively associated with disease. This is a reminder to prepare a lecture. So we can use all these tools together to start dissecting now these genetic associations, and we're going to focus on specifically the FTO locus, the strongest genetic association with obesity. What we're going to be seeking to do is understand and systematically decipher, number one, what are the relevant tissues and cell types underlying the disease? Namely, from this region of association, find the tissue and the cell type where that region is active through our epigenomic analysis. Number two, through this correlation analysis that I showed you earlier, establish the downstream target genes of these locus. Number three, through the regulatory genomics, establish what are the causal nucleotides that disrupt evolutionarily conserved regulatory motifs. Number four, who are the upstream regulators whose binding in these regions is likely disrupted by these SNP? Number five, what are the cellular phenotypes downstream of this? And number six, what are the organismal phenotypes? And we're going to be applying all of these to the FTO locus. So again, this is a paper in collaboration with Melina Clausenter and several folks that you probably recognize here. And this has major implications for obesity therapeutics, but deep down, we can think of this paper as a model for dissecting genome-wide association studies. So again, as I mentioned earlier, there are 89 common variants in this FTO locus. It is associated with obesity and also with several comorbid traits like type 2 diabetes and cardiovascular traits. There are 89 common variants. They span 47,000 nucleotides, and they sit in the first intron of this FTO gene. However, there are no protein altering variants, suggesting that there's probably a regulatory role, but leaving the target gene and the tissue of action a subject of great debate. So in fact, there are many genes in the region that are predicted to be the targets across different papers. So, there's different papers that claim that it was FTO itself, or IRX3 in pancreas, or RBL2 in lymphocytes, or RPG1L in brain, or IRX3 in brain, and so on and so forth. So extremely diverse set of predictions, completely inconsistent with each other. So how do we use the methods that I showed you to now start establishing the relevant tissue and cell type? Where, well, looking at the epigenomic annotations from the Roadmap Epigenomics project that I mentioned earlier, what we're finding is 12,000 nucleotide super enhancer spanning the first 10 kb of this region and acting specifically in mesenchymal stem cells. These are the progenitor cells that give rise to both white adipocytes, but also beige or bright adipocytes. In contrast to brown adipocytes that actually have more of a muscle lineage origin, these are extremely mitochondrial rich adipocytes, and these are actually your best friends. They're burning all these calories, whereas white adipocytes are actually storing excess calories as white fat. What brown adipocytes are able to do is thermogenize. So they can depolarize their mitochondrial membrane, causing leak of proton concentrations and loss of energy dissipated as heat. Now, beige and bright adipocytes have both developmental lineages as their potential progeny, and they can differentiate directly from these progenitor cells and also carry out thermogenesis, albeit to a smaller degree than brown adipocytes. So this is very tantalizing. It basically says that this is acting specifically in this developmental fate decision between white and beige adipocytes. So it's very tantalizing. And indeed, we found that there's a very strong difference in the enhancer activity of specifically the first 10 kb segment between risk and non-risk haplotypes. In the risk haplotype, which is associated with obesity, you see a de-repression of this enhancer and nearly a twofold higher activity, suggesting that that localization is probably relevant. Next challenge is to establish the downstream target genes. And to do this, we use three types of links. The first I already mentioned to you about, which is the correlation between the activity of the enhancer and the expression of the nearby gene across tissues. The second type of link that we used is the physical looping of the chromatin across distant regions. Specifically, if you look at this FTO locus right here, and specifically this genetic variant that we believe to be causal, you see this long-range interaction involving multiple genes nearby, but also that there are specific enrichments with IRX3 and with IRX5 in this locus. So you can see these two sharp peaks of activity, which basically reveal that the folding of the chromatin in that locus of the genome is bringing together these distal genes to the proximity of this genetic variant associated with obesity. And the third type of evidence is genetic linking, whereby instead of looking at the association between genetic variation and disease, we can basically look for an association between genetic variation and expression differences between individuals in the nearby genes. So we're going to be using expression quantitative trait loci to also show that the risk variant indeed causes expression changes in the genes nearby. So doing this, we're basically finding that indeed there are two large blocks of association surrounding this locus and 8 candidate genes within this region. And then looking at specifically homologous versus non-homologous, homozygous risk versus homozygous non-risk individuals, we basically asked, is there a genotype dependent expression? And what we found indeed is that IRX3 and IRX5, two genes that are sitting 500,000 nucleotides away and 1.2 million nucleotides away from this causal region, are in fact both showing a gain of function de-repression, specifically in the pre-adipocytes. And the risk allele is a gain of function. So that's something that is very common for gene regulatory variants, but much less common for proteins. Number two, this action occurs in early adipocytes. The EQTL is actually not visible in whole adipose tissue. This was only visible in the first 3 days of differentiation. So that's the second challenge of basically finding the target genes. And we now have pre-adipocytes and IRX3 and IRX5 as the targets. The third challenge is how do we find the causal nucleotide? And what we can do is carry out this analysis of which nucleotides are in fact overlapping these regions and which ones are evolutionarily conserved. So looking for a global enrichment, we indeed found that several motifs are enriched across multiple BMI GWAS loci, but also that one of them in particular within that first 10 kb region is in fact showing the strongest conservation of motif modules. Again, this is a method that Melina Klausnitzer established to basically look for not just motif level, but module level conservation across multiple species. And what she found is that indeed this ARID5B motif, which is this AT-rich motif, was disrupted in the risk individuals by a T2C alteration that abolishes this AT-rich motif, leading to loss of binding of this AT-rich interacting domain protein. And indeed, when you look at the difference in activity between risk and non-risk, you find that that difference is lost for a very small enhancer, but that's what we would expect because that enhancer was enormous. But you do see a very strong difference with increased binding, increased gain of expression for the risk loci. We then want to establish the upstream regulators. And indeed, what we're finding is that ARID5B, specifically among multiple ARID regulators, is in fact very highly expressed both in risk and in non-risk individuals. And indeed, when you do the epistatic analysis of the cis and the trans, where you're perturbing either the motif or the transacting factor, it is only when both are intact that you see repression. But if you disrupt either of them or both of them, you see de-repression, again suggesting that this loss of repression is due to an interaction specifically between that cis regulatory alteration and this trans regulatory alteration of ARID5B. And we see the same thing not just for enhancer activity, but also for IRX3 and IRX5 expression. We then turn to the cellular phenotypes. Great, we have the circuitry. We have the tissue, the target genes, the causal nucleotide, and the upstream regulator. But what does it actually do? What do these downstream genes, IRX3 and IRX5, actually do? To find out, we basically looked at the correlation between the expression of IRX3 and IRX5 across a large number of genes for every functional module in the genome. And what we found is that the two genes were positively correlated with lipid metabolism and negatively correlated with mitochondrial function. And indeed, this was also reflected in the cellular phenotypes, where the risk individuals showed negative, so basically showed less expression for mitochondria, less activity of mitochondria based on lower mitochondrial DNA level, and more adipocytes, larger adipocytes. So that suggests a shift between thermogenesis and lipogenesis. So instead of burning the energy in mitochondria, you're storing the energy in larger adipocytes. And that happens through this depolarization of the mitochondrial membrane, where UCP1, one of the major factors underlying this, is in fact the downstream gene that is controlled by IRX3 and IRX5. So the model that we basically built is this shift between the C allele and this T allele, between these white adipocytes and lipid storage versus beige and brown adipocytes and thermogenesis, where in this FTO locus, that single nucleotide change is in fact shifting between the two by causing loss of binding of ARID5B and then increased in expression of both the enhancer and its downstream target genes. So how do we now validate these organismal consequences? By having the circuitry, we can go in and intervene. We can knock out or knock down or overexpress the target genes. We can overexpress or knock down the upstream regulator. And we can even use genome editing with CRISPR-Cas9 to go and change that single nucleotide alteration. And what we can see is that each of these modifications is switching back and forth between lean and obese phenotypes. If you knock down IRX3, you can basically de-repress these, you can change the oxygen consumption rate from the risk individuals who are unable to thermogenize back to the normal levels. If you overexpress IRX3, you basically see that you take these normally thermogenizing non-risk individuals back down to the risk levels, and the same thing for IRX5. And if you go into mice now and you take normal mice and you push IRX3 not to higher expression to make them obese, but to lower expression to see if you can make them even more lean, what you can see is that indeed a dominant negative form of IRX5, which is specifically expressed in adipose tissue through AP2, is in fact leading to 50% of the fat mass ratio being lost in these mice. And these mice are now unable to gain weight in a high-fat diet. If you introduce a high-fat diet for the control mice, you see this gain in this gain in weight. And if you introduce a high-fat diet for these dominant negative IRX3 mice, you see no change in weight, suggesting that they are immune to weight gain. And that's because of increased thermogenesis, where they're burning weight, and they're burning calories when they're awake and also when they're sleeping. And then lastly, we can go in and edit that single nucleotide variant between T and C to basically go from the non-risk to the risk, and then go from the risk back to the non-risk. And what you can see is both IRX3 are changing in expression, getting de-repressed and repressed again, as well as thermogenesis increasing dramatically. Yes, the IRX3 mice are normally leaner, but the AP2 mice are normally leaner, but this change in weight is the main key result here. So they start out leaner, but they're unable to gain weight. So thanks for the great question. So this is all at the tissue level. We now want to go to the single cell level. And basically what we've been doing is looking across a large number of different disorders to basically systematically look for what are the changes that are happening in DNA accessibility and RNA expression across both risk and non-risk individuals, across more than 1,500 brains and across 7 million different cells and all of the major cell types. So what we can see here is this increasingly delicate and high resolution view of the mammalian brain and the human brain in particular for how are the expression patterns driven by these changes in Alzheimer's disease, and also that these changes are acting in all of the major cell types, changes in expression, which are also correlating with the sex of the individuals. The early changes in Alzheimer's are extremely cell type specific, but the late changes are much more shared between cell types. And there's a huge number of genes that are sexually dimorphic, that are very differentially expressed between men and women. And when you have Alzheimer's, this is an even more pronounced change in expression, suggesting, in fact, that we have to really be thinking about these disorders in a sex-specific and cell-type specific way. We're finding a lot of coordinated changes between single-cell RNA and single-cell attack in the Alzheimer's brains. And we're also able to call mosaic mutations that are happening during development and that are in fact affecting specific pathways in excitatory neurons and oligodendrocytes, which are associated with disease. We've scaled this to many, many different disorders. We're able to do this for schizophrenia, where we can see what are the differentially expressed genes and the cell types and the pathways. What in ALS and FTD, again, finding a large number of differentially expressed pathways in each of these major tissues. In Huntington's disease, we're basically finding that these indirect pathway spinal projection neurons are losing their distinct identity from the direct pathway neurons and merging. And this is something that we see both in human and in mouse. And across different brain regions, we're finding that the expression patterns are in fact revealing very different cell states between the different brain regions of these cell types. We're also able to look at spatial information from the single cell profiles, where we can actually capture different sub-regions of the subiculum and the dentate gyrus and CA1 and CA2 within the hippocampus that are in fact associated with different association with Alzheimer's disease. And we're able to distinguish different classes of microglia, including synaptic microglia that play roles in schizophrenia, and immune and inflammatory microglia that are playing roles in Alzheimer's disease. I want to briefly mention the work that we're doing in collaboration with Lori, looking at the multi-tissue effects of both exercise and diet across both human and mouse. And this is a wonderful collaboration with an outstanding team of people. Looking at 300,000 cells across these tissues of interventions across both diet and training, we can basically start asking what are all of the cells and how are they changing by tissue and how are they changing by diet and exercise. And what we're finding is that, in fact, the expression of these genes captures both spatial and cellular state information, where depending on where you are along the small intestine, you can actually see different subpopulations of cells. We're also finding that training, in fact, specifically alters the stem cell state and the T cell subtypes in visceral fat. And you can see here that the training, in fact, reverses some of the effects of the high-fat diet and moves them in opposite directions. We can also see how exercise actually reprograms the expression of stem cells and immune cells that are basically changing in training. And some of those are in fact specifically reprogramming pathways that are associated with proliferation as well as with immune dysregulation. And what we're finding is that the training also reverses the within tissue interactions that are induced by the high-fat diet. So we're able to now start predicting which cells are interacting with what other cells based on the set of receptors and ligands that they're expressing. And what we're finding is that the training reverses many of these interactions, that 78% of the communications between skeletal muscle and wide adipose tissue are in fact induced, that are induced by high-fat diet, are rescued by exercise training, providing a mechanism through which this effect of exercised muscle has across multiple tissues in the body. So we're also doing a lot of work in cancer and in identifying biomarkers of aging and blood biomarkers of disease, as well as the progression of disease across different treatments in the context of cancer. And we're also able to distinguish not just one phenotype, but multiple phenotypes together and how they're associated with specific combinations of these subphenotypes and how they're demarcating different subsets of cell types and different subsets of functional categories that are enriched in specific combinations of phenotypes. And we've expanded that across the electronic health records to basically start learning combinations of phenotypes that define different types of individuals, and how we can use that to recognize the cell types that underlie specific combinations. Lastly, we developed a lot of methods for high throughput dissection of these predictions by looking at the effect of activating enhancers or repressing enhancers using dead-Cas9 with P300 or CRAB, the effect of editing individual nucleotides and knocking out either promoters or genes in induced pluripotent stem cells, and then differentiating those in neurons, microglia, astrocytes, oligodendrocytes, and any other cell type, to basically start observing these effects in a cell type specific way. We've also developed a lot of methodologies for high throughput dissection of these regions by testing 7 million elements at a time through these massively parallel reporter assays and these self-transcribing reporters that allow you to insert any kind of construct downstream and then use it as its own reporter, enabling us to cut out accessible regions millions at a time from the genome, insert them millions at a time, and then test their effects millions at a time. And then lastly, we're collaborating with a former postdoc of my lab, Andreas Penning, to start testing tens of thousands of these genetic variants directly into the brains of mice to understand how are these genetic variants acting in the context, the endogenous context of all of these additional cell types in the brain. So that's where I'll stop, to basically tell you that we've talked about how we can use reference epigenomes to predict disease-relevant tissues, how we can combine genetic and epigenetic and a transcriptional variation in the context of disease, how we can study disease at the single cell resolution across both epigenomic and transcriptional variation, how we can mechanistically dissect these loci by inferring the cell type, the enhancers, the causal variant, the upstream regulators, their target genes, revealing this metabolic master switch in the downstream circuitry of RX3 and RX5, involving thermogenesis, and then testing it with editing and showing both cellular and mouse phenotypes. And lastly, the work that we're doing with Lori on the multi-tissue convergence of these changes in metabolism, but also in other tissues, and the multi-phenotype dissection and the high throughput dissection. Again, amazing set of collaborators. Many of them have actually started their own independent positions. We're always looking for postdocs. So please come join us, and I will stop there and be happy to take any questions. We do have 9 minutes left for questions.
Well, thank you, Manolis. That was a really fascinating talk. He joked before that he was talking to was going to talk till 7 o'clock tonight, and I think you had enough data on those slides that you actually could have done that for us. So maybe I'll start by asking a question, sort of a bigger, broader question, and then some of the questions will come in on chat. But so it sounds like the MWAS is more predictive of disease than GWAS. Is that what you were saying? Is that your?
So the main takeaway from that point is that there are, in fact, basically, this is an enormous gap to spread. And basically, there are thousands of genetic variants that are influencing disease. But every one of these thousands of variants is influencing disease by a minute amount. By looking at these intermediate phenotypes, we now have fewer variants affecting any one enhancer, but also fewer enhancers affecting any one gene, and fewer genes affecting any one intermediate phenotype, and fewer intermediate phenotypes affecting disease. So in any one of these layers, you're breaking up this extremely polygenic gap and finding stronger effect phenotypes. So that's why the association is stronger with MWAS or with expression or transcriptome-wide association studies, because you've reduced the number of variables, and these variables have individually stronger effects. Does that make sense?
Yeah, but methylation can be changed by environmental factors also, right? So doesn't that add another degree of complexity to this?
So basically, when we're looking at MWAS alone, then of course some of this variation will be downstream of disease. But when we're looking at imputed MWAS, then that is again only the causal component. So we can actually play them between the two. We can basically say, what is the genetic component and what is the residual to get directly at the environmental effects? You can basically say, if I subtract the genetically driven component of methylation, then what I'm left with is either noise or environmentally driven. And then I can ask, is that component now associated with disease? And perhaps some of that will be downstream of disease, enabling us to actually get the two sides of the arrow uncoupled.
Okay, great.
Why don't you read the questions?
Okay.
I'm going to be jumping through.
So, George, do you want to unmute yourself and ask a question?
Thank you for the great talk. Can you hear me?
Yeah, very well.
Oh, thank you. So wonderful talk. It's incredibly exciting, and I think, you know, I learned a great deal. I'm just wondering, have you had a comparative analysis between what we're finding in animal and human, since you're going across a huge different tissues and enhancers and so forth? So, because one major problem we have in a lot of metabolic diseases, what we're finding in mice model, I would say a majority of time actually cannot be translated into human. I understand you starting with human traits, so that's great. But can you bring some light to why there's such a big difference translating metabolic diseases from animal rodent, a mice model to human? For example, in the complication of diabetes, 95% of the time they don't translate. So that was the problem.
Yeah. No, I completely agree. And there's been, for example, the Nelson study showed that if you take drugs that have a genetic basis, then you have a twofold or more increase in the fraction of them that actually succeed. So the approach that we take in my lab is always genetics first. So we start with genetics, and then anything else we find, we know that that locus is already associated with the disease. We know that however minuscule the effect is of any one of these loci, we know that that nucleotide alone still has a detectable effect at the phenotypic level. And when we have that confidence, then we know that whatever biology we dissect surrounding this will have fewer opportunity for feedback that actually wipes out these effects. Basically, you can think of a mouse model as, yeah, sure, there's a lot of feedback mechanisms that maybe are non-existing in mouse, but humans have additional of these feedback mechanisms. So you reduce the diet or you increase exercise, etc. There might actually be feedback. But by having a genetic locus, you sort of see that the feedback mechanisms of the cell and of the body are not sufficient to overcome this one nucleotide alteration. And therefore, when you go in and intervene at that locus surrounding that circuitry, based on the full circuitry that you then elucidate, you have the ability to truly change that, that nucleotide. So basically, when you have that full circuitry here, you can actually say, yes, because I know that this one nucleotide has an effect all the way down to obesity, if I understand the circuitry, I can intervene to a much greater extent than what that one SNP can do. For example, by changing the expression of the downstream genes, I can actually have much greater ability to manipulate these phenotypes. Does that make sense?
Thank you.
So yes, I mean, huge challenge to go between human and mouse, but having genetics first allows us to have much greater opportunity and probability of translation.
Okay, thanks. We got just about 3 more minutes. So Ron, you had a question. Are you, was that answered by Manolis?
Ron's question.
Yeah, he did get my question, but Tiago, a fellow in my lab, has a question. Why don't we take that?
So Tiago is asking, are the long-range SNP genome interactions necessarily confined to a single chromosome, or could they be interchromosomal? So this is actually very exciting for me that you asked that, because with Leandro Agudelo in our lab, we're basically now showing that these topological domains that are actually underlying obesity in many cases are in fact forming these loops that involve multiple chromosomes together. So if you're interested in sort of going after these sort of multi-chromosomal interactions in these topologically associated domains that underlying obesity, this is something that we care a lot about and we've been working on for a few years now. So Tiago, it's a fantastic idea. It's something that we care a lot about. And there's a lot of evidence that indeed interchromosomal interactions are causing these domains that are coming together. There's a lot of work by Young Lab and others in sort of non-transcription factor mediated mechanisms of compartmentalization within the cell. And we do find that these involve cross-tissue and, sorry, cross-chromosomal interaction. So fantastic area of research. I see one more chat window question.
Okay. Ron, do you want to?
Yeah, if there's time, I'd like to. It was a beautiful talk, Manolis, and covered many things. You didn't comment or maybe comment at all about the non-coding RNAs and where do they fit in this regulation and in this linking between genetic loci and final biological effects?
Yeah, so it's a fantastic question. So link RNAs is something that we and many others have spent a lot of time studying, and we basically have had several papers on link RNAs and their specific roles. What I have to admit is that biologically, they're an extremely exciting and phenomenally interesting phenomenon. But if you look at their actual effects compared to transcription factors and other sort of these master regulators, most link RNAs are extremely weakly expressed, and sort of they're thought to serve as perhaps in some cases local scaffolds in sort of establishing the interactions between chromatin and all of the regulators that bind chromatin. So it is still a matter of great debate as to sort of how central are link RNAs to sort of the global control and circuitry of mammalian and human regulation. In our experience, they haven't been as clear slam dunk phenotypes as some of the transcription factors. So we've kind of shied away a little bit from that, but I don't mean to put the field down in any way. I feel that there's a tremendous amount to be learned still by link RNAs. It's just that they have smaller effects in our observation, and of course, much weaker expression than transcription factors.
Okay, great. Well, Manolis, thank you again for an outstanding talk. If anyone has any additional questions, feel free to email him. I'm going to tell everybody that. I hope that's okay, Manolis.
Of course.
And thanks again. Everyone, keep your computers on, see what's going to happen over the next couple of days. We're all hoping for the best, I think. So
I put my email address in the chat window, so feel free to email me, and I'll be delighted to collaborate. I mean, we're extremely collaborative as a group. And Lori's an amazing collaborator, but I'm sure that there's room for a bigger family too. Awesome.
Okay.
Thanks.
All right. Have a good day, everyone. Thank you.
Really appreciate it. And thanks for the kind invitation. Bye, everyone.
Bye.